Hello, Andrew Hebard here with the Archiflower channel, Regenerative Omegas for a Regenerative Planet. And today I'm really pleased to be here with Dr. Adam Metherel, Assistant Professor at the University of Toronto within the Department of Nutritional Sciences. Adam, thanks so much for joining us today.
Absolutely, thanks for having me.
So today is going to be a deep dive into science, and I hope I can keep up with you. It's not my forte and my area of expertise, but this is such important work that you're doing, and your findings are really quite profound in terms of the areas that we want to talk about in terms of omega-3 nutrition. So just to set the scene, before taking on the role of assistant professor, you did your master's and doctoral degrees at the University of Waterloo in Ontario in the Department of Kinesiology. And I had to look up kinesiology just to make sure I had my facts right, which is the scientific study of how the human body moves, I think.
Yep.
And there you were working on developing a rapid blood test for assessing omega-3 profiles. And from there, you did two I guess you call them fellowships, the latter of which was with the University of Toronto, working alongside Professor Richard Bazinet, who was an earlier guest on this podcast. And now you lead a team looking at if I understand this right, looking at the interplay between sex, genetics, and diet, and how those three things come together to influence the metabolism and biosynthesis of omega-3 polyunsaturated fatty acids.
Yep, that was an excellent summary.
Oh, good. Okay, well, we're together so far. So why I'm so really interested in the research that you're doing is that for most of my career in looking at plant-based omegas and different plant species that produce them, I've been living under the assumption, because this was the accepted belief, that plant-based omegas do not convert very efficiently in humans through to EPA and DHA. And we're at a time now where the omega-3 industry is facing some very, very big challenges around the supply, the continuity of supply of marine-based omegas, and the pricing increase that they're seeing and how that's translating through to the consumer. And if I look at the work that you're doing, it's using groundbreaking technology, very sophisticated technology, which we'll come on to, and having some really quite profound results, which are potentially upending this 50-year notion that we've been living under about this sort of innate inefficiency that humans have. So humans have as it relates to converting plant-based omegas through to EPA and DHA. So could you explain a little bit about what got you into this science, this area of research? Because you're using, I talked about groundbreaking technology. If I understand, I've got my terminology right, that is carbon isotope ratio mass spectrometry to follow the metabolic fate of fatty acids when they're ingested through to their sort of final destination. Is that broadly right?
Yep, that's right.
And what got you into that? What was the draw that got you into that initially?
It was about 6 or 7 years ago, probably, and while I was a postdoctoral fellow with Dr. Bazinet, who you mentioned earlier, we learned, and this was well known in certain research communities, but we learned that different plants actually can have very different carbon-13 levels, which is very much dependent on a couple of different photosynthetic pathways that each plant can have. So put simply, some plants like corn, sugarcane that are very abundant in our diets, they actually have a high carbon-13 content. Other plants like flax and ahi flower, they use a pathway that results in relatively lower carbon-13 levels. So, and then marine sources, things like fish, tend to have this more intermediate carbon-13 level. So we made that, we learned that, half a decade or more ago. At the same time, I was actually just completed an animal study that was feeding either alpha-linolenic acid or ALA from a flax source compared to a ALA combined with docosahexaenoic acid, or DHA, which was from fish. So from this, knowing what we had just learned essentially about the different carbon-13 ratios in these two sources in the diet, we realized that we could potentially, by feeding both of these in the diet, start to answer some fairly complex metabolic questions. So the first question we had, just because we had these samples, we knew the differences in carbon-13 levels between ALA and DHA. We wanted to look into a process known as retroconversion. So when you feed DHA to rodents, humans, really any mammal, EPA increases, or eicosapentaenoic acid. This is an intermediary omega-3 fatty acid between ALA and DHA. So when you feed DHA, EPA goes up, and this was long for 30 or more years now. It was assumed or believed that this was due to a process known as retroconversion, where DHA gets converted back to EPA. So we wanted to investigate this, and using the technology, somewhat surprisingly, we pretty quickly refuted more than 30 years of research in the area that suggested that retroconversion was the major pathway contributing to this increase in EPA. What we actually found was that it was EPA metabolism was actually slowing. So its conversion to downstream products was actually being slowed, and that was causing EPA to accumulate. It wasn't an increase in this retroconversion pathway, which does still exist, but it wasn't responsible for that increase. And then we were able to confirm this in humans. And this has actually opened a whole other avenue of research projects using in vitro, preclinical, clinical models, all of which are pointing us in this direction of this idea of really a negative feedback pathway where DHA is able to inhibit its own synthesis. So it's really opened up all kinds of different research streams.
So how does that feel as a researcher to develop a technology, an analytical technology, come up with a study designed to sort of employ that technology to challenge 30 years of prior science, and actually show that 30 years of science has been either misinterpreted or misunderstood? That's I would think that's a researcher's dream, isn't it? To sort of challenge the status quo, and that's what science is all about: is to continually push the barriers of our learning. I should think you and your team are pretty chuffed with that.
Yeah, on one hand, it's extremely exciting. We love kind of discovering new mechanisms, new pathways. On the other hand, this is 30 years of research that's kind of been embedded in the whole research area around the world. The big challenge is trying to convey and communicate the idea that what we thought we knew for 30 years is essentially incorrect, and it is a real challenge. We're trying to go to conferences and present this to the appropriate research societies, but there's still papers coming out where they're contributing the rise in EPA due to retroconversion. So it's exciting for sure, but there's a lot of work to try and get that message across.
I can imagine. So I'm going to go back a little bit just to make sure I understand this. When we talk about carbon-13, because in the world of fatty acids, we often talk about carbon chain length and C18s and C20s and C22s and 24, and we're not talking about that at all with carbon-13. We're looking at I think a unique signature or almost invisible fingerprint that these molecules carry with them that enable us to look at DHA and say there's two variants of DHA. One variant of DHA that would have started out life in the plant kingdom and one variant that started out life in the marine kingdom. So we can actually turn around and now look at how plant-based ALA or SDA coming from flaxseed or archiflower seed oil actually goes through, and which molecules of DHA it ends up in. So we can sort of do almost like a mass balance of how much ALA and SDA is being converted through to DHA. Am I thinking about that right?
Yeah, absolutely. It's very similar to what you would do with a stable isotope study,
right?
Where you might infuse an animal with a labeled ALA, or give a human an oral dose of a labeled, so a stably labeled ALA molecule. The difference here is we're doing this at a natural abundance level. So we're potentially able to avoid the need for expensive commercially made tracers, but it is essentially a stable isotope study where we can track metabolism, where that ALA or SDA stearidonic acid molecule is going in the body.
Right. Wow. So now I'm going to come on to the bit that this gets really, really exciting. Now, for me, it gets really exciting. So your most recent research involving archiflower, you did a diet switching model, and you did this in mice, and you looked at evaluating flaxseed oil, archiflower oil, and marine or purified DHA from fish oil. And your results, I believe, show that archiflower oil biosynthesizes naturally to form new DHA in key tissues with comparable efficiency as pure marine DHA. And I'm going to repeat that again, because each time I read it, I think, wow, that is upending 50 years of maybe conventional belief and science. And it is that archiflower oil, when fed to these mice, biosynthesized naturally to create new DHA in key tissues with comparable efficiency as marine DHA, which would have been unheard of a few years ago. You'd almost had that dismissed, say, no, I mean, it doesn't. All of our research would argue that, because it wasn't using this new technology that you're using. So it really is challenging this dogma that plant-based omega-3 sources are converted inefficiently to DHA. Again, am I looking at that right from your study?
Yeah, I think that's a fair assessment. I mean, when you talk about the 50 or so odd years of dogma, I mean, related to poor conversion to DHA from these plant-based omega-3 fatty acids, historically, usually ALA, but in the case of archiflower oil, you have the ALA and SDA. One of the really major supporting pieces for this is for inefficient DHA synthesis has been the fact that when you supplement these precursors, so ALA, SDA, even EPA, when you supplement those in rodents, humans, pigs, all kinds of different mammals, you don't see a change in DHA levels.
In the plasma, in the red blood cells or plasma?
In red blood cells, plasma, tissues, if you're looking at rodents. So you can feed up to, you can feed up to 14 grams per day of ALA and not get a change in blood DHA levels. That's 10 times higher than a normal human intake of ALA. So, but when we're only looking at fatty acid levels, it doesn't tell us how much is going through that pool. So presumably, if you feed more ALA, although it doesn't change DHA levels, there's probably more going through it, which is just being matched by downstream DHA metabolism. So DHA levels are at a steady state in this case, but by no means is it static. There's always something going in and something going out at the same time. And so I think really that's what this archiflower study has done, is it's been able to challenge that long-held belief that we are inefficient DHA synthesizers. And so we showed similarly when we feed ALA and SDA in our study that's under review, or actually under revision right now, we didn't change DHA levels when feeding ahi flower oil, or actually didn't change DHA levels when feeding ahi flower oil or flaxseed oil. And we were feeding them at levels comparable to what a normal human intake would be. But what we did show is, though, even though we compared this to a DHA fed animal, there was no statistically significant difference in the rate that DHA was being turned over, as you said, in a number of different tissues, plasma, adipose, liver, brain, compared to ahi flower oil. So certainly that and indicates that ALA and SDA are, in this case, a viable method for maintaining DHA turnover. But it's just a starting point, right? This is one study. We're going to keep working at it. And it's critical that we continue to drive this method forward into human clinical trials to try and assess how these plant-based precursors are able to turn over DHA in human populations.
Does that raise a question then, that instead, so you've demonstrated that ALA and SDA can convert DHA with comparable efficiency, but not increase the level of DHA, but it converts with comparable efficiency to pure DHA. Does that, put another way, maybe raise the question that it's not about how efficient we are, it's about how much we need, and we can make as much DHA as we need from plant-based precursors?
Maybe, yeah.
Maybe.
It's such a hard question to answer. The idea of how much DHA we need, no idea, right?
Yeah.
But
no, that's still to be determined. But it's it's really interesting findings. And going back also to this, the research that has shown that even increasing significantly the amount of ALA that we take in the diet, you didn't see a corresponding increase in circulating DHA. It's a little bit, as you say, it's the speed of turnover. And I guess it's a little bit like I was having a plumber explain to me about the flow of water, and said, well, there's two things. It's the flow and the velocity kind of water through a pipe that matters, and rather as to how much water comes out at the end of the spout. I know I haven't done that justice, but I can visualize this with turnover in the blood, that it's that velocity of change that is important, or it's showing, the science is showing us that it's that velocity of turnover that's important. And does that in turn mean maybe that just looking at plasma levels of DHA and EPA as a proxy for overall omega-3 health, there's more to it than that? So when we look at the omega-3 index, for example, there's more to it than look at just as the finite levels of EPA and DHA in our tissues as in our blood as a proxy for overall omega-3 health.
Yeah, for sure. I mean, the omega-3 index and a number of other biomarkers they're great for kind of identifying, usual, EPA or DHA intakes over the previous, months or weeks, depending on the biomarker you're using. And, there's, I think, as we all know, the omega-3 index has been linked to a number of different indications for risk of, cardiovascular disease or other health outcomes. But you're right, it can't capture really the metabolism of it. How much is going in and how much is going out, and therefore how much is actually being utilized by the tissues themselves. And I mean, you think about plasma, there's a finite number of spaces where DHA can go in the plasma. So if more is coming in, then more has to go out naturally.
Right.
So certainly, I think all the biomarkers like the omega-3 index have a lot of utility, but we're really trying to push towards having a more complete understanding of how those levels, when they're changing, how is that actually affecting the input and the output, or the overall metabolism of those fatty acids.
So when you say, it's interesting actually, I hadn't thought about this before, but when you say if more is coming in, must be going out because there's only certain places it can be stored, is that alluding to the fact that if we, through higher levels of supplementation of EPA and DHA, there's only so much that we can use sort of metabolically as a biologically active fat, and the rest becomes a source of energy and sort of is expired because we can only use so much of it for accruing in tissues and membranes and nerve sheaths and that sort of thing?
Yeah, I mean, I don't want to, this would be fairly controversial, but, if in an extreme, example, if you supplement EPA and DHA on a daily basis, 2, 3, 4 grams per day, maybe, your, EPA and DHA levels obviously start to go up as a marker of intake. In an extreme situation, maybe that increase in levels of EPA and DHA and your red blood cell membranes and your tissues could be indicative of a reduced need to metabolize it in your tissues. So once you reach a certain threshold, EPA and DHA consumption kind of plateaus, but there's still more of it coming in. It's possible that now you just need somewhere to put it, and it's just going into tissues and cell membranes and that sort of thing. I don't know that for sure. I have no evidence to support that yet, but it's certainly one possibility, right?
Add that to the list of needs further research and things. And so, because of the study that you did, and obviously the tissue that was being analyzed, it's very hard to do studies like that in human models. So they were done in a mouse model, and I'm wondering or interested to know, how accurately do you consider omega-3 metabolism in mice? Does that data translate? How translatable is it to omega-3 metabolism in humans?
Yeah, there's another kind of set of beliefs in this area of research that mice and rats are these like super converters or super synthesizers of DHA. However, there was a study, I want to say almost 10 years ago, that came out of Richard Bazinet's lab, where they applied the same models that have been used in humans to identify percent dietary ALA conversion to plasma DHA of less than 1%. That's kind of the generally accepted percent dietary conversion to plasma DHA levels at less than 1%. But they actually use these same models that have been developed in humans, and then went backwards and applied them to rodents, and found similarly, these values were also generally less than 1%. So based on those models, rodents may not be these efficient DHA converters, and might actually be a pretty decent model for humans. And so one of the follow-up studies that I've been working on is to really kind of get at this idea of, what's the percent conversion of ALA to plasma DHA. But because we're using rodents, we can also do a whole body analysis and look at how those plasma levels are actually really underestimating DHA synthesis levels.
Very interesting. And in an earlier interview, I was talking to Dr. Susan Kleiner, and she was talking about initiated pathways and used the example of lactose and lactase, and put it very simply, I suppose that we have metabolic pathways that if they're not used, the human body doesn't want to expend a lot of energy on maintaining these pathways if there's no need for them. And if we're not experiencing lactose in the diet, our ability to create lactase goes down. Do you see that with humans, or could we expect to see that with humans in different sort of categories of human? So I'm thinking particularly about vegans and vegetarians that may have a higher level of plant-based omegas in their diet. Therefore, they're using these sort of metabolic pathways with a greater degree of need, I guess, both need and also, maybe they're using a different balance of omega fatty acids in the diet, particularly thinking about omega-6 versus omega-3. Do you think you see, or do we see different efficiencies of conversion in vegan and vegetarian populations compared to sort of more flexitarians or carnivores, omnivores?
Yeah, I think there's a really good possibility for that. If you look at DHA levels of vegans and vegetarians, although they do have generally lower DHA levels than fish eaters or those who aren't vegans or vegetarians, they're not, drastically lower. So these are, populations that either consume very little DHA or no DHA at all, and they're still making DHA, and at a level that's comparable to other populations, occasional fish eaters even. So there's certainly that. I would hypothesize that they may have an increased capacity for DHA synthesis. And there's even, there's been evidence from Inuit populations that consume very high levels of DHA and EPA. If you go in kind of the reverse direction, that they are inefficient converters of, ALA to EPA and DHA because they don't need to, right?
Right. Yeah, good point.
So there's some really interesting stuff in that area of research, and again, another potential gap that warrants further investigation.
Yeah. So that is interesting. I hadn't thought of it quite like that. So, and another area of your research is that I think you've shown that EPA in the human body forms a pool or a reservoir for brain DHA formation. And it goes from EPA to DHA from this reservoir, even in the presence of high background DHA supplementation. So one thing we know from ahi flower, one of the many things we know from ahi flower oil metabolism, is that SDA gets converted very efficiently and very effectively through to EPA. So do you think ahi flower oil in that sense, which boosts circulating EPA, could also serve as a sort of functional alternative to fish oil in that respect, that it boosts this pool or this reservoir of EPA for onward conversion to DHA without the necessary supplementation of DHA?
Yeah, I mean, it's certainly possible. I mean, in the current ahi flower study, we're generally seeing higher EPA levels in the ahi flower fed mice. As you know, this has been shown in human studies as well, right? So on the surface, if ahi flower oil can increase EPA levels more than, you know, other plant-based omega-3 oils, there's certainly a possibility that you know, increase in EPA can more efficiently provide DHA to the brain. However, I just want to be careful because that previous study where we showed that EPA was a fairly substantial pool for brain DHA, it wasn't infused. So it was directly infused into the circulation, so it didn't have to go through the whole digestive system. And so it was certainly more readily available for metabolism. And at the same time, we weren't, because it was a secondary analysis of a previous study. So we unfortunately weren't able to do kind of a head-to-head comparison between EPA infused and DHA infused animals to really kind of say, you know, EPA provides this amount of DHA for the brain, preformed DHA provides this amount. They're either comparable or they're not. We couldn't do that. So there's certainly some questions on how we can do that. But, you know, nevertheless, we've in the current ahi flower study, although, you know, not knowing how long we needed to do this study heading in, although it ended up not being long enough to fully capture brain DHA turnover, we are seeing DHA turnover rates that are fairly similar between the ahi flower oil, and actually statistically similar between the ahi flower oil group and the DHA fed group. So it's certainly, you know, pointing us in that direction, that, you know, ahi flower oil might be a reasonable source for brain DHA metabolism. Again, you know, we need to maybe move forward with some more studies that can actually directly compare, you know, ahi flower oil versus EPA versus DHA in a properly designed study that has enough time points that we can really capture brain DHA turnover, because it is very slow and requires a much longer study design.
What is the typical recognized as the turnover rate of DHA?
From preformed DHA, we've multiple different models. The diet switching model has shown this. Some of the infusion models, short term, like 5-minute, really quick infusions, intracranial infusion of labeled DHA as well. They're all coming in kind of in that 30 to 45 day half-life of DHA.
Right.
Whereas some of the plant-based models have kind of been landing in that 80 to 100 day range, particularly from ALA alone.
What questions does that raise in your mind about why one source versus the other one would have such a, well, basically twice the half-life?
Well, I mean, when you feed preformed DHA, it doesn't need to go through nearly the number of steps that ALA has to go through to become DHA, right? It's you consume it, it's absorbed, digested, absorbed, and it's kind of there, it's ready, it's quickly available for, you know, brain DHA uptake. If you're consuming just ALA, it needs to be absorbed the same as DHA does, but then it has to go to the liver, be converted to, you know, elongated, desaturated, all these different steps to be able to be converted to DHA. One of the other factors for ALA as well is that a large portion of what we do consume for ALA on first pass actually gets taken up by adipose and stored.
Mm-hmm.
And then, so, you know, maybe 3/4 of what we eat actually goes into adipose fairly quickly. Another 3/4 of what we don't store in adipose actually becomes beta oxidized. So in the end, we also have probably less ALA available to even be converted to DHA in that case.
Right.
So there's a few different things that could be going on.
Interesting. Well, there's a lot more there to unpack in future conversations, future studies, but I did want to sort of finish on, I'm very mindful of your time. What I start out by saying, I can't imagine there are many researchers that get with using the new technologies that they've, or new analytical technologies that they've developed and design studies around it, apply them to research that challenge such a deep-seated, almost taken as granted scientific basis in an area so profoundly important as health and nutrition. Like there's a lot of areas I think you could do this in, and it would be, well, that's interesting, but it doesn't maybe touch the lives or the planet in such a profound way as the work that you're doing. And I think the impact that you're having, the research that you're shining light on for human health and planetary health, as we think about the importance of omega-3 nutrition, which is, they're indisputably one of the most important nutrients that our bodies can take in, and that we also recognize we're not getting enough of them. We realize that we have to look at sustainable, replenishable, regenerative sources rather than depletive sources and destructive sources. So your work in that area is very important. We appreciate it greatly. What's next on your bucket list of areas to research? That kind of, we're painting a picture here, but there's clearly gaps in there. There will always be gaps. We fill one gap, another gap appears. But what's on your sort of short-term bucket list, Adam, of the things that you'd like to sort of pursue in this area of advancing our current knowledge?
Yeah. Before I get into that, you kind of touched on an interesting point there, that there's other molecules that, we could apply this technology to. And I think that's really important. It's not just about fatty acids and omega-3 fatty acids. Really, you could apply this carbon-13 mass spectrometry methodologies to any different molecule that you can have a slightly different carbon-13 signature for. Sugar comes to mind, right? Corn and sugarcane have those photosynthetic pathways that give you a high C13 or carbon-13 level, while beetroot sugar, which is also fairly common, more so in Europe than North America, has a lower carbon-13 content. So you can actually do this with sugars, which of course, as we know, is also very important nutrient in our diets. So yeah, just want to kind of highlight, it's not all about fatty acids, but for me, it is.
I hadn't thought about that, but the metabolic consequence and the nutritional consequence of cane sugar versus, as you say, beet sugar would be fascinating. I hadn't even contemplated that there would be a difference there, but yeah, why shouldn't there be?
For sure. But yeah, as far as like what's coming up, we have some really exciting publications coming up where we're looking at DHA synthesis and turnover rates from dietary ALA, dietary EPA, and dietary DHA in male and female mice. So we're using the same diet switching model that we used for the ahi flower study. And the preliminary data is really, really convincing that we are better DHA synthesizers than previously believed. This model is giving us that 1% value, or less than 1% dietary ALA conversion to plasma DHA value that is so cited in the literature.
Right.
But when we consider all the other tissues in the body and really look at it on a whole body level, we're actually looking at about 13% of dietary ALA is being converted to DHA in the body. So if you consume 1,000 milligrams of ALA a day, 130 milligrams of DHA is being produced. So that's more than 40 times higher than what we would have previously thought if we're just considering plasma levels alone.
That is amazing. That really is interesting. Again, it goes back to just how much we've got to rethink the current metrics that we're using and draw conclusions from those metrics. And this statement about, we're all innately inefficient, which clearly doesn't make any sense from an evolutionary point of view, but the work that you're doing really shines a light on that. That is very, very impressive data, and something, again, to really build on that, to show that difference between 1% and 13% is huge.
Yeah, and I mean, we're always going to have these issues of trying to translate these numbers to humans, right? I mean, we're looking at it on a whole body level in a mouse. Really hard to do that in a human, right? Really, we have blood to work with. We can sometimes, maybe we'll get an adipose biopsy or a muscle biopsy, or something like that. But, looking at it on a whole body level is really difficult. That said, one of the other really exciting studies we have coming up is applying this model that we've developed in mice into a human population. So we are in the process of getting this study up and running. We're going to look at DHA synthesis rates in young, healthy, male and female vegan and vegetarian populations. So these populations that are low DHA consumers, and so might have a higher need to be able to synthesize DHA from ALA. Mm-hmm. And so the goal of this study is really twofold: is to be able to translate these animal models into a human study, and then to look at any sex differences in DHA synthesis rates that could explain higher DHA levels that we often see in young females compared to males. And so we're kind of looking at sometime next year to be able to start recruiting participants with that.
Wow. Well, Adam, it's fascinating. I could talk all day and ask you questions all day long about the work that you're doing, but that really is a great overview of your current research work that's ongoing at the moment. And I know our listeners are going to be thinking that is having profound effects on how we think about, the conventional, and I don't want to use sort of terms like dogma as it relates to this, but the conventional thinking about plant versus marine omega-3s and the impacts that they may have. So thanks so much for sharing that with us. And we look forward to following your research and continuing to collaborate with you. And all the best for your ongoing studies and taking those items off your bucket list.
Thank you very much. And it was a pleasure being on.